Hi everyone, I am Tahir Abdulrehman. I am an MSc eHealth student at McMaster University. Our poster is entitled Data-Driven Computational Drug Repositioning for aureus Associated Diabetic Foot Ulcer. The advisors of this study are Dr. Shamir Khader from Imperial College London and Dr. Kamlesh Yadav from the School of Engineering Medicine at Texas A&M University. So coming to the introduction, diabetic foot ulcer is a complication of diabetes mellitus and is usually characterized by wounds or ulcers in the lower limbs. The most frequent pathogen associated with diabetic foot ulcer is Staphylococcus aureus. Due, due to antibiotic resistance and the persistent nature of the pathogen, it poses serious challenges to conventional treatment methods. Aureus infections associated with diabetic foot ulcer can lead to recurrent infections, and in severe cases, it may even lead to amputation of the associated or affected limb. So our objective is to identify potential therapeutic drugs for aureus-associated diabetic foot ulcer using a computational drug repurposing approach, utilizing gene disease association methods or analysis and biological pathway enrichment. So coming to the methodology, so we utilized DisGenet, which is a comprehensive database of disease gene associations. So utilizing specific keywords like diabetic foot ulcer and Staphylococcus aureus infections, we were able to identify uh, the genes that were associated with aureus associated diabetic foot ulcer. So once we identify the genes through this genet, we utilize another web resource enricher, and using enricher we conducted a functional enrichment analysis of the KEG 2021 human biological pathways to identify human pathways that were associated or linked to this genet identified genes. Also, we conducted an enrichment analysis of the IDG drug 2022 database to find or enrich drugs that could be potentially be repurposed for aureus associated diabetic foot ulcer. So we have given a flowchart of our methodology in the poster. So coming to the results, so we have identified 398 genes utilizing this genet, and these genes could provide insights to the existing approved drugs that can be repurposed for aureus associated diabetic foot ulcer. So the enrichment analysis of the KEG 2021 human biological pathway database uh, helped us to identify 132 pathways with an adjusted p-value of less than 0.05. So these human biological pathways that were enriched from the KEG 2021 database helps us or gives insight into the pathways that could be modulated for drug repurposing approaches. So the top four pathways that were enriched from the KEG 2021 database were IL-17 signaling pathway, the cytokine receptor interaction pathway, tuberculosis, and rheumatoid arthritis. So table 1 it shows us the details of the top four human biological pathways that were enriched from the KEGG 2021 database. Also, we performed an enrichment analysis of the IDG drug 2022 database, and we identified 48 potential drugs with an adjusted p-value of less than 0.05. So among the 48 potential drugs, the top three significant drugs were quercetin, sorafenib, and disulfiram. So these drugs could potentially be repurposed for aureus associated diabetic foot ulcer. So table 2 shows the top three drugs, as well as their associated genes. So in conclusion, computational drug repurposing methodology could help to identify human biological pathways that could be modulated for drug repurposing approaches, or or these computational methods could help to identify drugs that could be repurposed for aureus associated diabetic foot ulcer. But further studies like in vitro, in vivo, and clinical trials are needed to validate the potential therapeutic effects or uh, beneficial effects of these identified drugs. Thank you for listening, and if you have any questions regarding this study, you can send your questions to the corresponding author. Thank you.